Please be seated. Good afternoon. Good afternoon, everyone. A warm welcome to each one of you for the grand inauguration of SVATV, celebrating the success of GenomeIndia Project. It is indeed an honor to stand before such a distinguished gathering today, as we embark on a four-day journey of knowledge, innovation, and awareness. SVATV is not just an event; it is a celebration of science, progress, and reminder that experience beyond borders. To commence this auspicious occasion, let us stand in unity for the national anthem. Thank you very well. Kindly be seated. As knowledge illuminates our mind, we begin this program with auspicious Saraswati Vandana and the traditional lamp lighting ritual. I now invite our esteemed dignitaries to kindly step forward for the lamp lighting ritual.
ब्रह्माच्युत शङ्कर प्रभृतिभिर्देवैः सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजाड्यात्मा
Thank you for gracing this moment with your presence. Dr. Mahesh Bedekar, Dr. Vrinda Mazrankar, and Dr. Kavita Muriman and Jayashiman to please come on the dais. We also have Sri Vidya ma'am with us. Before we proceed, let us extend our heartfelt gratitude to our esteemed dignitaries through a token of appreciation. I request Dr. Vinda Mazamkar to present a token of appreciation to Dr. Mahesh Bedekar, sir. Next, I request Dr. Kalpita Mule to present a token of appreciation to our principal, Dr. Vinda Mazamkar. I request Dr. Kalpita Mule to present a token of appreciation to Sri Vidya ma'am. As we embark on this journey of discovery, let us remember the words of James Watson, the co-discoverer of DNA. The greatest legacy we can leave behind is a deeper understanding of our own biology. The GenomeIndia Project is a transformative step toward understanding the genetic diversity of our nation in healthcare, precision medicine, and as well as in disease prevention. Today, we stand in the frontier of science where every gene tells a story and every discovery shapes the future of human health. To officially commence today's proceedings, I invite our principal, Dr. Vinda Mazrankar, to deliver a welcome address.
नमस्कार. राम राम. उठले सगळे? So I welcome management member Dr. Mahesh Bedekar. It is his brainchild and he told us we have to do this because it's a massive thing which is happening and it is targeting all our Indian population, that is focused only the India as an Oriental, and we are having a vast diversity, and it is this efforts that which was brought to shape by Kalpita madam, Jayasri madam, and I am a little part of it because genetics is at my heart. And whenever somebody teaches genetics and teaches something wrong, then I intervene. That this is not this. Because I've been teaching this for a couple of years, like 25 years I'm into genetics, teaching genetics. So I know the pedigrees, I know the how to assemble the data and the sex link and the autosomal and the point mutation. So whenever you take me up, I will wake up. So that should be the genetics. And now there will be good jobs for them to become a genetic counselors. You know, these are the good jobs now. It's a good field which is coming out to be a precise medicine and all those things. See, you must have seen on the Amazon. Nowadays they tell you that we will give you customized face packs and hair oils and all. So it is slowly coming out. So you can see there are big giants like Novartis, Piramal's. I think they must be working, but it can be a corporate secrecy. And then slowly, once maybe 3-4 years, they will come up to your house and say, yes, give us your sample and we will give you precise medicines at your doorsteps. So that can happen and that is really going to save your life. And thanks to the one who has started this GenomeIndia project, that is the scientists who have worked efficiently with the low cost. You know, we always Indians are known for it, notable for it. If our autoclave is not used working, what we do? Pressure cooker. Right? We do it. So we know how to get the things done and we don't wait until this has to be happening. And that is what is the Indian brains. Alright? So we have to appreciate ourselves and pat our backs that yes, we know the solutions for the problems And that is why most geniuses who are operating in the US market as a giant are all Indian brains, right? So we want no the brain drain now to happen. It should be settled in India, and we should be a big power. And that's all I would like to talk about the genomics. That we are the best genes accumulated from north and south and southeast and west and the what do you say the Filipinos and the Eurasians and the Huns and the Shakas, Caucasoids and Aryans, and we are a beautiful admixture of everyone. So definitely we are different and we will be different, and that is what we should. Thank you so much.
Thank you, ma'am, for your inspiring words. Ladies and gentlemen, we are privileged to have amongst us Dr. Mahesh Bedekar, an esteemed BPM member and the visionary behind SWATV. It is his foresight and dedication that have made this event possible. I now invite him to share his thoughts.
Good afternoon, friends. Principal Madam, coordinators, and friends. Madam gave a brief introduction, and this was about, I think, 2 months back. And I just read an article in the newspaper, I think in some of our medical journals, that GenomeIndia Project has successfully completed some sequencing of genes. I was knowing about this project maybe about a 1.5 years back, but it didn't struck my mind. When I read that article, I felt that I was so naive about this project. Myself being a doctor, I am a pretty medicine expert as well. I see genetic problems which are happening in the screens of couples which come to me. But I was naive about this. As a doctor also, I was not knowing about it. I knew about the project, but somehow it had missed. Then it came into my mind that we have an education institute. And common man, forget about common man, specialists who are working in this field, like us or maybe other doctors are also not knowing in depth about this. We need not know about in depth about the genetics, but at least as a nation, we should know that what massive work we are doing. So it came in my mind, and we have a biotechnology department, we have a very biotechnology department. So I requested them that we have to do something on this matter. And in no time, such a wonderful work has been done. We had just to tell Madam and the department, rest everything, what you see here is their hard work and of course students behind that. And what I envisioned should be a project or it should be an exhibition where common man will understand what is this project. We don't want to tell about what it's a very difficult subject where common man will get demotivated. If you start telling them about chromosomes and everything, so naturally they will not like it. But telling in their language by street and doing models. So such a nice exhibition. These all are biology students and engineering also. So I thought, what is my ecosystem? My ecosystem is this college. So let's start with our college, where science college as well as all other our sister institutes. Like we have law students, Madam is there. We have engineering college, we have arts commerce college, we have management students. So their students also should know about what this project is and what as a country we are doing. So we are a country which recently we are celebrating Kumbh Mela as well. So we are a country where there is a spiritual journey which is going on more than, they say more than 50 crore people there participating. And we are a country, the same country is achieving milestones like going on moon and doing such type of work. So such diversity is not seen in any other country in the world. So we as a citizens, it is our responsibility to go about this country. These scientists, they are our real heroes. They should be our role models. It is said that there are no role models which are there in our lives. Students don't have role models. It's not the fact, it is our job. There are lots of people in our country who are role models, but they are not projected. They are scientists, they are working in their own labs, they are in their company. They don't want any publicity. Those who want publicity, we know them, what they are. They know how to do good PR. There are agencies, there are ways to do it. But these people, they don't know about it and they are our role models. Any country is only progressing because of them. And what we are different? We are different because Europeans did it long back. Americans did it long back. But somebody in India thought about it. Government gave proper support, proper funds were allocated, and you see the project. This project is going to change overall. Human Genome Project is going to change the way medicine is going to be in the future. Not only medicine, agriculture. There are so many vast applications for this. You know, in USA, breast cancer is a very common cancer. Cancers are not that common. Let's not scare everybody. But in females, the commonest cancers which are seen are breast cancer and cervical cancer. But there is no vaccine for breast cancer. We have a vaccine for cervical cancer. But there is no vaccine for breast cancer. And in American black women, the incidence of breast cancer is very high. But unfortunately, there is no proper screening, and the amount of females coming forward for screening is also less because of their problems of black women and their socio-economic status. So they have identified a gene, which gene can be modified, and if you give proper guidance to this woman, there are more than 40% chances of a black woman. If you compare a black woman and an American white woman, the black woman has more than 40% chances that she will have a breast cancer. So that breast cancer, they used to come to the doctors when it went to second stage. So now because of such type of projects, they have started identifying such those communities, black communities. The gene is identified. That is a BRCA1 and BRCA2 genes. That means if you have a history, if your mother or a first degree relative has it, the next generation has to check for those genes. And then there can be some preventive measures by which such type of, because breast cancer is a very dreaded disease. By the time the patients come to us with second stage of breast cancer, so screening is very important. Still, it's a long way to go because gene mapping is done, sequencing is done, but these are not screening tests. What is a screening test? Screening test is a test which is very easily done, which is very cheap and universally accepted. Unfortunately, it's a long way to go. Even abroad, these tests are not affordable, only a certain strata of population does it. But in coming time, and when countries like India go into it, then the cost reduces. We are allocated some 238 to 50 crores of rupees, but US took, they had the budget of $3 billion. There is no comparison. The same work India can do it with not one third, minuscule resources and amount. So when countries like India come into it, where we know how to cut costs and give this type of service to common man, because still it is not a screening test, it has to be a screening test where all strata of society can do it. As on today, a select strata of society who can afford this can do it. But there is, in coming times, I'm sure, when we are going into it, it is going to change the way we do it. Personalized medicine, as I told you, the very good thing which Human Genome Project told us that we all are equal. Only 1% difference. Can you imagine? All human beings are equal. That has been scientifically proved. Nobody can question that. But there is a 1% difference. That 1% difference has been researched. And because of that research, so many new things are coming. But which is scientifically proved is what is important, that human race is equal. Social factors change, and that 1%, when good social factors are incorporated, that 1% also those people who are susceptible for certain diseases, if they are social, there are social, because black women, their social economic conditions are weak. the speeding is weak. That social changes in the chain, they can be equated with biochemistry. So all these things are very interesting. India, why India is important? Because majority of the data which we have is from the West. The population is different. That 1% difference is also huge. So Indian society is diverse. You know that. We all know that every kilometer language changes. Every kilometer the sex changes. So this data was very important, and this data is going to help the whole genome project, not only for India but the whole world is going to help. And this is an ongoing project. In coming times, I'm sure much more progress will come into this. Colleges like us, departments like us, it is our responsibility that our students and the society should know what good work we are doing. With this, the nation, because the best investment for a nation is health, nothing else. Education and health, these are two things. There should be no GDP. How much amount of GDP is spent on health and education? If you ask me, government should stop counting that. Because the more you invest into health and education, that is the one which is going to help me, our generation, and our future. Thank you so much.
Thank you, sir, for your valuable insights. A short video would be displayed now of all the glimpses from today's morning. the hard work of the students involved. It is now time for much anticipated keynote address by none other than Dr. K. Thangaraj, the National Coordinator of Genome India. Thangaraj sir is presently a CSI Bhatnagar Fellow at the at the CSIR Center for Cellular and Molecular Biology, Hyderabad. He was director of the Center for DNA Fingerprinting and Diagnostics, Hyderabad from 2019 to 2022. He was also director, additional charge of the Institute of Stem Cell Science and Regenerative Medicine, Bangalore, during February 2022 and August 2022. His main research interest for the last 3 decades has been in the field of population and medical genomics, and he has published about 300 plus research articles. He is an elected fellow of Indian National Science Academy, Indian Academy of Sciences, National Academy of Sciences. He is a recipient of several awards including J.C. Bose Fellowship, Sun Pharma Research Award in Medical Sciences, Raman Research Fellowship, Lifetime Achievement Award, Excellence in Science, and delivered several ovations. He is a board editor of Mitochondrion, associate editor of BMC Medical Genomics, Tropical Medicine, and International Health. He is also member of Indian Society of Human Genetics from 2011 to 2015 and founder, also the president of Society for Mitochondrial Research and Medicine. He is going to enlighten us on the topic population genomics and public health. Also, I would like to make an announcement. After the guests has completed their presentations, attendees can submit their questions in the following ways. The online attendees can post their questions in the YouTube comment section, and offline attendees can write down their questions in the circulated. Dr. Thangaraj, please enlighten us.
So, very good afternoon. Could you hear me?
Yes, sir. Yes, sir.
Okay. So, let me share my slide and see. You have to give a permission for me to share the slide.
Yes, sir. Now it is.
Is it on full screen mode? I'm trying to put it on full screen, but it's not.
Sir, you can go for slideshow. There could be an option at the bottom right corner.
No, this is not giving that option.
You can go for even F5, sir. F5 if it functions on your laptop.
That's okay. I was trying there. Is it okay now?
The slideshow is yet not visible, sir.
Not visible. One second. Let me first put this.
Sir, if you can again share the screen.
Yeah, I do that. I do that.
Sir, on the topmost there would be a slideshow option wherein there is a home tab.
After sharing, it's not showing.
Near the home tab, sir, top
No,
Left corner,
I was
there is file home.
trying, but not.
Slideshow, there is a slideshow option just next to animations.
Yeah, there are multiple options, but I'm unable to. That. Here is the one who's not taking.
Sir,
Here is another one who's not taking.
sir, at the top there is file, then insert home, insert design.
No,
Yes, here you can see slideshow, sir. Slideshow.
yeah, I could see, but
So click on that.
no, it's not taking. No, because already it's.
Can you press F5, sir? F5. Function F5.
Is it okay now?
So can you mail your
No,
slides?
no, this is a lot of.
Okay, okay.
Don't go. Is it showing now? Let me rejoin. Otherwise, right? I'll exit and rejoin.
Sir, I hope you are sharing the entire screen tab when it is asking to share.
The entire screen is not coming.
Uh, is the slideshow visible on your system, sir?
Let us try now.
Okay, yes, yes. Now it will be visible. Yes, sir. Yes, now it is visible, sir. Yeah.
Okay, right.
Thank you.
Okay, so sorry for this small change. First of all, I must thank the organizers for organizing and celebrating the success of Genome India project. Even we have not done so much that we have done for celebrating the success of the Genome India project. As all of you are aware, we have just completed the 10,000 genome from 83 different Indian populations. I'm audible, right?
Yes, yes, sir.
Okay. So I guess that most of the participants are students, right?
Yes.
So let me begin slowly with what is genome, and I'll give you some perspective that why Genome India is necessary. So then I'll come back again to talk about Genome India, right? This is the structure of my talk. So genome as a biology students, can you please mute mic, all others who are online? Because some noise is coming. So genome, as you all aware, that it's a complete genetic information which is present in the cell. So it could be a nuclear genome where that comes from the chromosomes, or it could be a mitochondrial genome which comes from mitochondrial DNA, which we all inherit only from the mother, whereas the nuclear genome is equally contributed by both the parents, father and mother. But what Francis Collin, who was sequenced the very first time the human genome from NIH, he has different kind of definition to say, so what is the genome for a lay person? He says, genome is a shop manual with an incredibly detailed blueprint for building every human cell. He also say it is a history book, a narrative of the journey of our species through time. And this is more important that he says that it's a transformative textbook of medicine with insights that will give healthcare providers new powers to treat, prevent, and cure the disease. So this is what, in brief, what Francis Collin said about. Then the question is that there are several millions of genome worldwide is available. So what is the need for Genome India? So why the Genome India is very important. But if you look at this picture, whereas the other countries or continent, if you see the people, they all look similar in terms of their skin color or features. But look at Indians, every single population is very, very unique, right? You have, even if you take color, you have very light color to very dark color. So that gives that India has very huge amount of genetic diversity, and therefore whatever existing genetic knowledge or information is not really useful for India because we are genetically very unique, right? So to give you some idea that how the Indian populations are stratified, that's mainly because of the stratification, the variations which we have in the Indian population, all because of the stratification and practice of endogamy over thousands of years. The Indian populations are stratified into four major linguistic groups: Dravidians, Indo-European, Austroasiatic, and Tibeto-Burman. They also now, every single linguistic groups, you know that there are several languages. For example, if you take Dravidian language, there are four languages: Tamil, Malayalam, Kannada, and Telugu. Similarly, all other linguistic group has several languages. But under every linguistic group, uh, I could hear some background noise. Can all please mute the mic?
Yes, sir. Yes, sir. Still, it is. There is disturbing noise, sir.
Yes, yes, some noise is coming.
Okay. Okay.
Maybe others who have joined online also should mute the mic. Now
Sir,
it's
is it a background noise of fan or some, or you are getting an echo?
no, not echo. Now it's better. Now it's better.
Okay.
So the every linguistic group has several social groups, so-called upper caste, middle caste, lower caste, tribes, and primitive tribes. So that makes actually India as one of the very complex nation in terms of the population diversity. So for the last 30 years, we have been trying to understand the history of the population, why such a huge diversity exists in India. So what is the genetic relationship between different ethnic populations, and so why the genetic basis for several diseases in India are very unique. So these backgrounds are very important for conceiving the Genome India project. In this process, to our surprise, we came to know the very first out of Africa human is in India. You all know that the modern human arose in Africa about 200,000 years back and migrated out of Africa and populated in the rest of the world. So while doing our research, we came to know the first out of Africa human is in Indian subcontinent. They are none other than the tribal populations of Andaman and Nicobar Islands. I don't know how many of you are aware. In Andaman and Nicobar Islands, there are four tribal groups who typically look like Africans. They are Onge, Jarwas, Great Andamanis, and Sentinelese. I'll not go into detail. Maybe some other occasion we can talk more detail about this population. We had a chance to go and collect the samples from the tribes, particularly this is the tribe called Onge, of course, several years back. Initially, we sequenced the complete mitochondrial DNA. In the beginning, I told in the genome we have nucleonome, which is contributed by both the parents. We have mitochondrial genome, which is contributed exclusively by mother. We all carry the mitochondrial DNA, which was inherited only by mother. In order to trace the maternal lineage, we use the mitochondrial DNA. If you want to trace paternal lineage, of course, the Y chromosome is only present in the male, so we use the Y chromosome markers. In this case, We sequenced the complete mitochondrial DNA, and that gives evidence that the very first modern human who migrated out of Africa, which taken southern coastal route and reached Andaman and Nicobar Islands, are the tribal groups of Andaman and Nicobar Island. They are the first modern human who migrated out of Africa and reached here about 65,000 years ago. Of course, so this is very old study, 20 years, published in Science those days. So this is the first genetic evidence suggesting the first modern human who migrated out of Africa is in Indian subcontinent. Subsequently, when the human genome was sequenced and the variations all along the human genome was identified, which are polymorphic across the world, and from there 1 million genetic variations, particularly the SNP, single nucleotide polymorphic markers, have been identified, and that is available in the form of chip. So in the next study, I'm just highlighting only few studies, we used the nuclear marker, 1 million genetic markers, and we selected 25 different population groups, again representing all the linguistic group, representing all the social groups, and we came to know that so there was two distinct populations in the prehistoric India. One, we call them as ancestral South Indians, called as ASI. They were part of the early human migration. That means the Andamanese migrated from Africa. That's the deep-rooted divergence. While migrating, taking southern coastal route, some groups stayed in southern part of India, whom we call as ancestral South Indians. There was another group called as ancestral North Indian. Probably they migrated much later from Africa towards north, and somewhere in the Middle East they diverged. One group has gone to Europe, other group has come to northern part of India, whom we call as ancestral North Indian. So why we kept this very close is there are some markers, including the markers which the mutation which changed the skin color from black to white are common in Europe, Middle East, and also northern part of India. Similarly, the lactate persistence mutation also common in Europe and northern part of India, but not in other parts. So that's why we have placed that these two are very close, and these two are the very first people who have settled in Indian subcontinent. First ASI, then ANI. Of course, there are several couple of other um, uh, linguistic groups also came much later. On the other side, if you look at this, is called as principal component analysis. These are all different Indian groups. These three are the HapMap sample, haplotype map of the world. This is Europeans, Africans, and Chinese. Where here you see that there are some hundreds of Europeans are there, hundreds of Africans, and hundreds of Chinese. They're all in single cluster, suggesting that they are genetically more homogeneous, not much different. Therefore, they are in single cluster. Whereas look at Indians, every single population is very unique. Although every single population is unique, but still there is some kind of genetic affinities exist among these Indian populations, right? So that makes very interesting that every single population is unique, but still we all have connected genetically, right? At the same time, we also shown that there's an admixture between ancestral South Indian and ancestral North Indians, and the present day populations are combination of ancestral South Indian genome and ancestral North Indian. If you go to extreme north, you will find maybe 80 or 90% of ancestral North Indian genome and maybe some 10% of ancestral South Indian genome. Similarly, if you go to extreme south, you have vice versa. So I'm not going to detail again, but in order to find out when this admixture took place, we took another study where we took large number of population from south and large number of population from north. Based on the recombinations using very strong statistical tools, we have shown that initially ancestral South Indians and ancestral North Indians have gave rise to many populations independently. You can imagine that the initial settlers have settled in a jungle or forest, and that group must have divided into two groups. One group must have stayed there, other group must have migrated to other place in search for food, and from there again people must have migrated. If the migration is several hundreds or thousands of years, naturally when we acquire mutation, every population acquiring mutation remains in that particular population. Right? So that's what makes everybody is very unique. Then this happened until the last 4,000 years. For the last 2 to 4,000 years, these two groups have admixed. The color indicates there's a kind of mixture of the genome between these two groups. This happened 2,000 years. After admixing, every single population started maintaining the endogamy. Endogamy means marrying within the group, right? So this itself suggests that initially two founding population, they gave rise to many population groups. Then there's admixture between these groups. Now for last 2,000 years, every single population started maintaining the endogamy. So now if you look at every single population, because of marrying within the group, so they acquired unique set of mutations, which makes every single population unique. But whatever happened here, this is what connecting every single population. And even the previous slide where the PCA showed there's a connection between each population. Why these are strictly maintained might be because of the caste system which arose background. Now I am coming to GenomeIndia Project. This is the very first meeting which we all, at some of us, attended, held in Indian Institute of Science. Professor Vijayalakshmi was the one who have driven this initially, and some of us are here. Christopher Balakrishnan, co-founder of Infosys. Professor Anurag was the director of Indian Institute of Science. I'm here, and some of the other scientists are were there in the meeting. The idea of having GenomeIndia Project is to have whole genome sequence of 10,000 individuals representing various linguistic and social groups across different parts of India, and create an exhaustive catalog of genetic variation of different level, common variants, low frequency variants, very rare, single nucleotide polymorphism, and also structural variants, and construct a reference genome for Indian populations. If somebody have very low coverage sequence, there's a missing data, they can use, impute, and get the missing data using the appropriate reference panel. And design a genome-wide array, because when we do genome sequence, because of the background which I mentioned earlier, ours is one of the oldest population. Every population is maintained endogamy, acquired unique set of mutation. We are expected to have large number of variation. We already found more than 130 million variations. So some of the variations can be used in the existing array so that we can have much better representation of the variations of Indian population for the future genetic studies. So that will be useful for precision and preventive medicine, not only in India, but also outside. Another important aspect of this is to create a biobank for both DNA and plasma sample. So that would give a lot of opportunity for researchers to make use of those biobank sample for future work. So these are the 20 institutes all over India. And you can say, for example, if you take Telangana, there are 2 institutes, CCMB, CDFD. If you take Tamil Nadu, IT Madras, and similarly, there are several institutes all over India. 20 institutes are participating there. And we assigned, or those who have, want to participate, they themselves have asked for specific work on this mission program. Some centers are exclusively for collecting samples. Those are represented in blue colors. Some are developing the methods. Some collected the sample as well as sequence. Right? For example, there are 4 centers: CCMB, NIBMG in Kalyani, IJM in Delhi, and the Center for Brain Research in Bangalore. So these are the 4 sequencing centers. 2,500 samples each have sequenced. So this gives you idea about what are the institutes involved in this study. And this gives you idea that which are the place where the samples have been collected. The dots represent the place where the samples have been collected. There are about 70 different locations. And as I mentioned, that it represents all linguistic groups and social groups, and also different geographical regions. So this, in fact, last year, exactly 1 year back, 27th February, has been announced, where there's an announcement in Delhi. Honorable Minister of Science and Technology has announced about the completion of this 10,000 genome. The time it was completed. Very recently, January, you must have heard, and also you are celebrating that, that has been released for public use. Honorable Prime Minister came on video conferencing and has actually talked about the importance of GenomeIndia data, and it says that it will help in developing precision medicine. There are a lot of write-up about that, and after 1 year, we had an extensive analysis. Right now, the 10,000 genomic sequence are being archived in Indian Biological Data Center at RCB in Faridabad. That's one of the DBT institutes. Now it will be available for researcher to make use of this. So anybody want to use, they have to follow a strict protocol. And recently, DBT has also called for a proposal where anybody want to make use of this can apply for that, and DBT will give fund. Of course, it's very, very competitive. They have to write a project such manner that it's compete with others who are submitting the project. One can also directly write to the committee giving appropriate reason for accessing this data. And initially it's available only for the researcher, not for any companies. So that way DBT and DBT secretary is very, very keen on giving it to the researchers. Therefore, the researcher can benefit out of this mega project because of huge amount of money spent on this. So this is in between that I'm highlighting initially what is the need, now what we have done, and now I'm showing just one example that how I'm connecting that, how our finding and how uh, GenomeIndia data is useful for that, right? So now initially, as I mentioned, that this is a disease which has very, very low frequency in a particular population. But what is the distribution of this mutation around the world? When we looked at the Indian GenomeIndia data, 10,000 individual, none of the population, none of the individual in any of this 83 population carrying this mutation. So it's clear. We also looked at 8,62,000 individuals genome, which is available for the public to use. We found only 2 individuals carrying that mutation in heterozygous condition. We could not find who are they, but they are South Asians. right? That means this is very rare mutation. But if we go to the population, it's highly prevalent. At least we identified 10 families who had a history of such children born and died within 2 months. Now I will take you through this, that what are things we have done and what we are going to achieve right now. And this is the novel mutation which we found is not been reported so far. Therefore, we wanted to know biologically what is the function of this mutation. It could be simple association, but not every individual who carries the mutation have the disease. In order to assign a function of this mutation, we created the animal model using CRISPR-Cas9, precisely deleting this 11 base pair, and we found that the pups are not surviving beyond 2 days. The children survive only for 2 months, whereas the pups are dying within 2 days after the birth. But we could still see the blisters, which is on, which is coming after 1 day. So the pups dying in 2 days. Unfortunately, we could not do any further experiment with this. Fortunately, we also created another one, another deletion, 45 base pair. This is the 11 base pair deletion, the region, and this is the 45 base pair deletion. While the 11 base pair deletion uh, truncates protein and it removes about 385 amino acid, whereas this is not truncating immediately, but it removes only 15 amino acid. Therefore, the impact of this, although deletion is much bigger, the impact is mild. We created an animal model for this also, and we found until 4 months there's no sign of any such skin disease. But after 4th month, they slowly start showing the phenotype what we have seen in the child, right? And after 6 months, it dies with a severe phenotype. This is a wild type, this is the complete mutant. You can see the size is so small and with so much of the phenotype with skin rashes, they die. But when we looked at the internal organ, this hypertrophy, spleen is enlarged, stomach is very small. We also had an opportunity to look at the skin biopsy. This is a wild type, it's intact dermis and epidermis, whereas here you can see it's very loosely arranged, and also there's a gap, that's where the blister starts and the skin rashes all comes. Then we also looked at the micro CT because the heart is very small. In the 11 base pair deleted mice, which die, the pups dies in 2 days. We looked at the heart and we found that you look at the cross section. This is wild type, this is heterozygous. One chromosome is normal, one is abnormal. These two look similar. Whereas in the 11 base pair deletion homozygous, the heart, if you look at thickening of the intervertebral septum, and also you can see its closing is called as hypertrophy. It also show a septal defect. There's a hole between the septum. This is a normal, right? There's a hole. So all this causes because of the 11 base pair deletion. Therefore, the animal is not surviving, and also the children are not surviving. So with this, what we can further do as a responsible citizen, responsible scientist, whether we can help the family. So in the meantime, we found this a mother who lost already a couple of children because of disease, is pregnant, and we wanted to do prenatal diagnosis. We brought the mother to Chennai, collected the amniotic fluid, and tested whether the fetus is carrying the homozygous wild type, homozygous mutant, or heterozygous. This is a homozygous, this is heterozygous, and the amniotic fluid also carries the heterozygous. Therefore, we advise the mother to carry forward the the fetus and the pregnancy and the child born about a year back with the normal phenotype. That gives some kind of satisfaction that whatever we have done at population level, which we started about 3 decades ago, slowly understanding not only the population history, also understanding that there are certain populations are expected to have population specific disease. And we went back and did some analysis, and we got something to contribute to the society. Okay. So not only this, but now we wanted to go a little beyond this. So we approached the government to give us the permission to screen the entire population in a particular region. There are some 5,000 individuals. There are several procedures. So one has to approach the Tamil Nadu government and attached medical university for clearance. Then has come to now in the hand of public health department. It's almost approved with minor corrections, which was suggested by ethical committee. So once they approve, we go and screen the entire population. Based on the mutation, we'll give either prenatal diagnosis or premarital counseling or premarital counseling. Therefore, in the future, there will not be any child born with the disease. In fact, we can also eliminate the mutations after a few decades. So that's what is our plan. And if it works, we identify there are several other such diseases in several other populations. We want to continue that to other populations as well in the future. Therefore, we can contribute to the society. So in this line, when I was director of CDFD, we initiated a new program called Mission Program on Pediatric Rare Genetic Diseases. This is again funded by Department of Biotechnology, and there are 16 different institutes, as you see the dots here, are part of this, and they're all actively doing genetic diagnosis. And if they don't find any mutation, they send the sample to CDFD. CDFD will do either exome sequencing or whole genome sequencing, and based on that sequence, there will be a initially prediction tool to identify whether the mutation is pathogenic or have any impact. Then we use animal model to understand, as I've shown here for epidermolysis bullosa, to look at the impact of that mutation. Then we also try to treat some of the disease, particularly hematological diseases, and most importantly, we wanted to make awareness for the people who are who are who are needing such kind of thing, and anybody, even some of you come across any such diseases which are rare in nature, can help or can inform them that they can approach CDIP. And the website, somebody can look at it, the pediatric rare disease program. There are 8 different language. There are videos. One can easily understand the impact of that and how the mission program can help the population which are in need for such thing. So this is the last slide. With that, I would like to thank all those who have contributed. Without big contribution from large number of collaboration, will not be able to complete such large thing. So once again, I thank the organizers for and so I stop sharing the slide. I'll be happy to answer if you have any question. Thank you so much.
Thank you so much, sir. Uh, as we understand, this is a lot of massive amount of work that you have concisely put up on the slides, and it was very easy to go through your slides and understand the type of work We are very grateful to have you with us here today for enlightening us on GenomeIndia Project. I have a question, sir, from audience. India being so diverse in population, do you think that the results of Genome India project can be a miniature of human genome project undertaken globally?
Yeah, so considering the large number of population, more than 4,600 population, 83 is very, very small. So 1.4 million people, only 10,000 is very, very small enough. But this is a good beginning. At least we don't have anything so far. And this is a good beginning, and there's a lot of opportunity for us to do more such genome sequences in the future.
Yes, sir. Thank you so much. I also think, like while listening to you, I also thought that this could also lead to a truly made in India human being later with CRISPR-Cas9 understanding all human genes in Indian population. We can maybe we can have a designer of made in India human. Anyway, thank you so much, sir. I sincerely express my gratitude to our keynote speaker, Dr. Thangaraj sir, who, in spite of his very busy schedule, kindly accepted our last minute invitation for this event. Sir, your speech has not only provided valuable insights on Genome India, but also motivated young minds for career in this field. Thank you very much, sir.
Thank you. It's my pleasure again talking to you. And as you said that if students are inspired, I'll be really happy.
Yes, sir.
They are taking genetics as their research.
Yes, sir. Actually, we would also like to collaborate in this project, and if our students can be a part of this project in their later post graduation or PhD research, that would really be great.
I think there's a lot of opportunity, but of course not in this particular project. This is already over. So
Okay.
I'm sure that in future there may be several such projects coming up, and they are most welcome to join in that.
Sure, sir. Sure. Thank you so much.
Okay, thank you. So I'll leave from the.
Sure. Up next, we have Dr. Punit Prasad, Principal Investigator of Genome India, to share his expertise and experiences with us. A distinguished scientist with vast experience in molecular biology and genomics. Dr. Prasad has made remarkable contributions in the field. Dr. Punit Prasad works as scientist at Institute of Life Sciences, Bhubhaneshwar. He has obtained his doctoral degree from Southern Illinois University in the USA in 2010. He moved to Karolinska Institute, Stockholm, Sweden, in his postdoctoral studies. He has worked in the field of chromatin and epigenetics for more than 2 decades. During the COVID-19 pandemic, he has contributed immensely to COVID-19 testing and surveillance as a part of Indian SARS-CoV-2 Genomics Consortium by Department of Biotechnology. He has also contributed to the consortium project of Genome India. He has published several peer-reviewed articles in international journals. There are several postdoctoral fellowships and national grants to his credit. He is a recipient of prestigious Ramananda Swamy Fellowship from Department of Biotechnology, Government of India. His research interest is in understanding epigenetic mechanisms behind acute myeloid leukemia and importance of microbiomes in various disease conditions. Sir, we eagerly look forward to your insights.
Okay, Am I audible
Yes, sir.
Yes.
and visible as well, or
Yes, you are visible, sir.
okay, good. So I'll just share my screen now. Is my screen visible?
Yes, sir. Yes, sir.
Okay. Uh, Thank you very much for having me. And I mean, it's really interesting to see that the institutes and colleges are actually celebrating Genome India uh, success project. And I'm also very happy that you were able to hold one of the best and the eminent scientist, Dr. Thangaraj, to start this series. And nothing could have been better than this. So I really congratulate the whole team for putting together this whole program. And what I am going to do is slightly different than what Sir did. Uh, I would try to go a little bit lower in terms of the science. Try to make it slightly more simple for people to understand, and I'll discuss more about how this Genome India project would be beneficial. Okay, how it would help India in multiple aspects. And in between, what I'm going to do is to put some concepts, basic concepts, textbook concepts, just to refresh, and then connect these with the big project that we have achieved so far. So before I go, I'll just tell you, we Institute of Life Science is located in Bhubhaneshwar in Odisha. Yeah, in Odisha, and it's a small institute with 28 faculties. We have now over 20 PDFs and around 150 students. And we work in the area of cancer biology, infectious disease, plant molecular biology, and interdisciplinary biology. The institute was established by Odisha government, but then in 2002 it was taken over by DBT. Thankfully, this research institute has a very strong infrastructure as to whatever you can think of cutting edge technology or cutting edge machines or any infrastructure. This has everything. Okay, and we were one of the first DBT institutes to help the Indian government as well as Odisha government to start doing the testing. Okay, without having any more manpower, the faculties and the students and the staff, they worked together to do the testing, and then they did a lot of research as well on COVID-19. But today the talk is slightly different, and we will focus more on Genome India aspects and broader implications. So before I go, we'll discuss a little bit about what is genome, what is genomics. These are some of the terminology that we could keep on using it very often, but maybe we do not understand the integrity of that. And then complexity in studying these genomes. What are the different aspects, different genetic, epigenetic, non-epigenetic, environmental aspects that integrate and affect our genomes, and of course the phenotypes as well. Then very briefly about Genome India Project. Dr. Thangaraj already covered most of it. I'll just briefly go through. There will be some slides that are kind of duplicated, but then that's pretty much. And then most importantly, how these Genome India or Indian genomes that have been discovered, what are inside them, how these can be utilized for future implications, health benefits, and using the cutting edge methodologies and technologies. So genes versus genome. Genes are a specific bunch of sequences in the DNA that can be expressed into protein. However, genome is all the DNA that is present in the nucleus, right? Then what is a genome? Again, if you see from a cell, the nucleus, it is the chromosomes, and within the chromosomes there is a DNA, and DNA is made up of adenine, cytosine, thymine, and guanine. This is all textbook stuff. Okay, and genome of an organism's complete set of DNA. For viruses, even they are RNA, and it contains all the information that a cell or an organism need for its normal well-being. So if you can see, different organisms have different genome sizes. The smallest from viruses, and the largest we have the mammals. Okay, plants have much more variable genomes, and if you see on left side, the viruses is around 170,000 base pairs. Our bacteria goes to around 4.6 million base pair. Then Drosophila or fruit fly is 130 million, human 3.2 billion, and 150 billion is a canopy plant. So you can understand the the length and breadth of the complexity that these genomes of different organisms harbors. Now, what is genomics? Genomics is basically the study of these genes. Okay, so the chromosomes, the DNA, and what is embedded within the DNA. And how do you do that? This is by doing the sequencing, understanding what is the pattern of ATGC, what is the sequence of them, what is the abundance of them in overall within a genome, by doing in-depth sequencing and mapping them to understand what kinds of genes, proteins they harbor for. So in recently, the genomes have been sequencing primarily dominated by Illumina for short read sequencing, and you have Nanopore, Oxford Nanopore for long read, and PacBio also for long read sequencing, and different capacity of sequences can be generated every day using these high throughput sequencing machines. If you see the advancement of genomes, okay, which in 1980s where only 10 KB can be sequenced per day, and if you can see here, this is over 10 to the power 9, okay, or 10 billion bases can be sequenced. In fact, more than that now, due to the advancement of technology, the methodology, and the speed by which this sequencing gets generated. Contrary to this, you can see there is 100,000-fold reduction in the price of sequencing, okay, from 2000 to 2022, and now it's even cheaper now, and faster and cheaper. So more and more data is getting populated into the system, and what is the need of an hour is to understand this data, to understand what does this sequence means, how these sequences actually terminates into proteins, what are the genetic and epigenetic component into that, and how this can be integrated to understand the complexity of the genomes. So this is well versed. We have read it during our childhood, the central dogma, DNA to mRNA, and then polypeptide, or the transcription and translation, which actually leads out to the functional proteins. Now, this video, I will quickly show you, especially the students and undergrads, to just visualize the complexity of the DNA that we have and how it works.
The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. Next, the nucleosomes are packaged into a thread. The end result is a fiber known as chromatin. This fiber is then looped and coiled yet again. Leading finally to the familiar shapes known as chromosomes, which can be seen in the nucleus.
So what you just now saw the complexity of the DNA. It's very easy to think that you just sequence that, but there's like 100,000 fold. The DNA is wrapped. The compaction of this DNA happens to be embedded inside the nucleus of the cell. And imagine such compact DNA has to be opened and closed dynamically in order to do this gene expression, where DNA can be transcriptionally activated to make mRNA, and then mRNA to protein, and so on. And then this protein is synthesized in the cytosol. Now, beyond genome, you also have an epigenome. Okay, so epi means over it, on top of it. So which means that this chromosomes that harbors DNA, okay, has histones, and these histone tails are modified. Now DNA is also modified with methylation and so on. Okay, so it can be categorized as writers, erasers, and readers. What are writers? These are the enzymes which post-translationally modify these histones, erasers which remove them, and readers which read these modifications in order to perform a very, very complex gene regulation, including opening of the chromatin fibers. Okay, now these both genetics and epigenetics are actually inculcated on our day-to-day life, okay, as to which is also affected by the environment, okay, and which dictates the phenotype, which is phenome. Okay, so what you eat, what environment you live, and which is also dictated what kind of genes you harbor, which means that if you have the propensity to become an obese, or if you have or not, or if even a thin and lean person can have the high level of cholesterol just because they are carrying some of those genetic variants in their pedigree. So the Human Genome Project started way beyond 1990, when this was conceived in US and the project was started. And the first chromosome 22 was sequenced in 1999. Okay, during this time both the technological development has happened and the sequencing platforms have also improved, and finally to 2003 the complete genome of human was published. Okay, but this still had 97% of the genome as well, because there are the chromosomes contain a lot of regions which were not easily sequenced with the current technology, and therefore they are also called as dark regions or dark genes, which were not sequenced, which are now sequenced. So that's why you have an older version of genome and you have a higher version of or a newer version of genome, which is called HG38 now. So this is the slide that you already saw. We were around 20 institutes were involved, and we were primarily involved in sample collection. However, now we are also analyzing the data, the sequences from this consortia, as well as we have some other projects to find out the genetic diversity that is embedded in the human population. If you think about this, why, in despite of having genomes that is already available from US and all, why Indian genome was so important, right? So the Indian genomes, you can understand the diversity that harbors, and Dr. Thangaraj very clearly showed how North and South India are diversified, and how with different caste system and all, how mixing has happened, and more genetic diversity has been created in the Indian population, which means that there are genetic pools which are both unique as well as common, and we need to understand that. And later on my presentation, I'll tell you why they are important to understand. But you can see that most of the Indian diversity was still untapped, and with 10,000 genomes by Genome India, this is now opening up. The reference genomes have been made for different communities, different ethnic groups, and so on. and within that you will try to understand, what kind of mutations are there. If there are different kinds of mutation will lead to genetic disorders, or can lead to genetic disorders, and so on. During this time, Genome India has not only done the sequencing, but it has also done training and capacity building to different places, so that all the institutes have the capability to do sequencing as well as analysis, and so on. This is what we have done is, like, we have sequenced Oriya Brahmins 353. Then these are the different tribal groups of Odisha that we are sequenced. And this is more SC, scheduled caste, which we have sequenced. This is zero, but this number needs to be changed because we have now around 100 samples from Adia as well. So you can understand, the complexity from Odia Brahmins to tribal groups, just within the Odisha itself, we have done, and we see huge diversity among them. So this is again, when the Genome India was completed, it gives a snapshot in a minute as to what all things has been done and what needs to be completed. And this is what I will focus from slide onwards.
In a groundbreaking step, the Genome India project has been completed. Launched in 2020, this pioneering initiative aimed to sequence 10,000 Indian genomes, creating a comprehensive database showcasing the country's vast genetic diversity. Supported by the Department of Biotechnology, this multi-institutional consortium project focused on identifying unique genetic variations across regions and communities. Such insights can revolutionize healthcare by enabling precision medicine, where treatments are tailored to an individual's genetic profile, improving disease prediction and treatment outcomes. Beyond medical advancements, the project positions India as a global leader in genomics, contributing to the international understanding of human genetics. With India's bioeconomy growing from $10 billion in 2014 to over $150 billion today, initiatives like this are pivotal for sustaining innovation and growth. The Genome India project not only enhances our understanding of genetics but also sets the stage for groundbreaking progress in healthcare and biotechnology.
So what you just now saw, what are the multiple implications that this project actually harbors? Okay, and that's where the goals and impact of Genome India becomes a lot more important for us. Okay, you already know the overview what this project says. The objective was not only to understand genetic diversity but also to work on precision medicine, disease predictions, prevention, drug response studies, and so on. And of course, we both Dr. Thangaraj and I have been telling about why Indian genomics study is so important at this point of time. So what Genome India and what it could be done now is my subsequent slides and rest of my talk. Okay, as it said very clearly, by understanding the genetic landscape of Indian population, it will pave the way for personalized healthcare, improved disease prevention, better patient treatment strategies. However, the success depends on downstream application and so on. So what there is, as Dr. Thangaraj was saying, there is a call where the sequences are now available, but the sequences cannot be given just like that because of a lot of privacy and ethical issues. So there is a call for the proposal where in different categories, where one can make work on different kinds of reference genomes and see the diversity of reference genome based on the compared to the Caucasian genome, which we all use for all our research purpose. Precision medicines, if there are specific mutations embedded within a particular community, that can be where the medicines can be personalized. Epigenetic changes, the DNA methylations that could have changed, or that has differences within the communities. Okay, then population specific genetic risk factors, if the person having different kinds of disease propensity or not. And what are the different kinds of drug variants, okay, genetic variants that are present during for which is important for drug development. Of course, cost-effective screening or diagnostic tools, which would be more India specific. And of course, one can predict disease outbreaks, because if you know that certain population are vulnerable for certain kind of disease, and you know that such disease comes during specific period of time, then one can take, or the public health policies can be attributed towards, designing goals and responsibilities to take care of such infections and prevent further infections, right? So I have been using a lot of these terminologies, and I will slightly simplify by defining those strategies, and then showing you how Genome India aspects are important in all these different aspects that I've just told you. So we have to understand one thing, that we do not live in isolation, right? Our genome, our individuality is not only dictated by the genomes, but also the epigenome and the exposome. Exposome is nothing but the environment and the lifestyle. Okay, now the another factor that is coming is the microbiome, or the gut microbiome, which also affect significantly our genomes and the genetic variants, and so on. So why is it important for this such Genome India kind of projects, or even for India it is important for healthcare and research? You understand, genetic pattern, genetic compatibility test, genetic ancestry, which people have, or Dr. Thangaraj discussed. You can have fingerprint, you can have disease predisposed, and finally the personalized medicines. Okay, and then all these things can happen only if we know each and every detail of the genomes, or the individual genomes that is live within India. Okay, so hence Genome India project plays a very vital role in understanding all these aspects. So I have been using a term, very mutation, genetic variants, and so on. Okay, this is textbook, but just to revise your memory, what is mutation? It is nothing but a change in a genetic component, or the basis, or the sequence of those bases, where there is a normal protein and there is abnormal protein that is synthesized, and so on. Now, there can be different kinds of mutations. You can have substitution, okay, where one set is replaced by another set. You can have deletion, where certain one or more than one base, or genetic are deleted. Insertions, where there are new bases that are inserted, and inversions, where there is flipping of these bases or bunch of bases, which can lead to differences in the protein expression. There could be two kinds. One is germline, which is hereditary, which is carried from within the parents, and the other is the somatic mutation, which is not carried or inherited, but it happens due to your lifestyle or the environment that you live in. Okay, now genomic variants. What are genomic variants? These are the change, okay, of certain bases, which we call as the variants. Okay, there could be duplication of the genes, there could be duplication of chromosomes, there could be deletion, as you can see, there could be inversions, and there could be translocations. Okay, and all these things affect significantly, and decades of work has been dedicated to autoimmune diseases, developmental disorders, brain disorders, and metabolic disorders, and so on. Okay, just a few examples. You can see there's or what you call is um, uh, inherited disease. Okay, inherited genetic variants, which comes from the parents to all the way to the to their progeny, and you can see the differences in the limb defects are there, and they are inherited. There are other chromosomal aberrations, okay, where you have, it's called Down syndrome. You would have heard about it, where there is another chromosome 21, Okay, so instead of 46 you have 47 chromosomes. Then there is a cry of a cat, where there is both body and brain development, and the child cries like a cat. You have Turner syndrome, where the one of the X chromosome is missing. There is lot of other, both bodily or secondary sexual characteristics which are defective, and so on. So all these things we would be able to understand. Okay, another important or one of the example, just to uh, give you the importance of such in-depth sequencing studies, where you can see that you would have seen in videos or said that a person can walk on fire or on hot coal and nothing happens to them. So it turns out the study was done in Pakistan first, and they found out that it is nothing but there is a autosomal recessive trait. Okay, when autosomal recessive trait comes, the sodium pump, which is important for pain, uh, does not open in the nerves, and therefore the person does not feel pain. Okay, so basically there is nothing, no voodoo in it, but there is the science behind it. There is a genes that is mutated. It has a recessive character, due to which he does not feel pain, and people think that there is something new that has happened to him and he is extraordinary, but it's not. Okay, then comes the pharmacogenomics. Okay, I've discussed about the mutations, chromosomal aberrations, and so on, and all this can be integrated when we study in depth the sequences that we have obtained from Genome India, and so on. The another aspect, very important aspect, is about the pharmacogenomics. What is pharmacogenomics? Is basically pharmacological aspects and the genomic as to how it affects. You would have heard and see, this medicine works for me, but this medicine does not work for me. Okay, and somebody say, okay, the same medicine, same composition works for somebody, but it does not work for others. Another question is why? How does it happen? It happens because of the differences in the genome, differences in the genetic component composition. Okay, and this figure exactly tells you that if you have a heterogeneous individual, drugs that are not toxic but beneficial, drug that is toxic but not beneficial, drug toxic but beneficial, drug not toxic, not beneficial at all. You can understand how this there's different drug actions that can happen to an individual. So our current medication is what, if you have certain problem, a defined pathophysiology, doctors give you a particular medicine, and it is assumed that that medicine will have an effect. If it does not have effect, then the patient come back to the doctor and they say, it's not working, and the doctors then change the medication, and sometimes that medication work, or sometimes that also does not work, and the process goes on. So it is not about what doctor is prescribing, but what doctor does not know, that what is you have inherited, or what is the genetic component you have. Will you be able to, will this medicine work for you or not? That's where pharmacogenomics, or the personalized medicine will come, and that is the future of the medicine, as you can see here. Now the question is, how do you go about it? Okay, again, the aspects that Genome India has covered. You can see the population, you need to get data, you need to get treatment. As I said, there is a plasma and all these blood samples that are systematically stored. One can take those to understand different outcomes, and all these aspects can be integrated, not only the genomics, but multiple omics like epigenomics, pharmacogenomics, and a lot of different aspects to basically understand what and how these genes or the genetic variants embedded within the population will have a effect on the drug. Then another very important aspect that is coming and getting, you would have, all the children would be hearing about machine learning, artificial intelligence. Okay, now Google also give you some Jenny kind of stuff. I don't use it, but I'm just telling. And it just summarizes everything for you. The question is, can we actually use these methodologies to integrate or to understand and to better predict the genomes that we harbor? And this basically tells you why AI or machine learning in genomics is important. Because imagine the amount of data that you have millions and millions of sequences that is present. It is practically impossible for a human being to actually understand, so you need very high end computational algorithms to actually understand. Once, even if just the genetic component you understand, how will you integrate the familial component, lifestyle, environment, and the medical component? And all these are important for integrating the effects into the system, so that we understand what genes we harbor, why do we harbor, what benefits we have, and what harmful effects we have. Imagine if you know that you are having defects for cardiomyopathies, you will be much more careful. You will be taking medications from the beginning. If a person is aware that they are hereditary for breast cancer, the person can go at regular checkups and make sure that the disease or those cancers are tapped much before the onset and can be treated. Okay, so all these things aspects are extremely important, and this can be very well articulated using machine learning and artificial intelligence algorithms. Okay, now how basically it happens, any Facebook, Instagram, which everybody, all of you uses, they basically, what they do, they gather information. Okay, and the same system is here. We gather information. The more and more you give the information, the more is the better and is the training model. And then you can deploy those and do the much more accurate prediction, where genomic information can be very well decoded, uh, and then one can understand molecular mechanisms and understand disease effectivity and disease diagnosis. And this actually has multitude of effect and advantage, where you will have real-time data analysis. Of course, ethical AI development has to be taken care. Personalized medicines can be created, or the community-based personalized. So you see, the India has so diverse population, so much diverse ethnicity. Okay, so if you know that certain ethnicity are predisposed for certain disease, or then you know that these kind of personalized medicines or medications can be given to them. Similarly, enhanced drug discovery or personalized drugs can be given, and more genomic epidemiology can be studied and can be very well regulated and controlled for better lifestyle. Now, another important aspect that is coming is what we call as genomic counseling. Okay. Now, what is genomic counseling, right? Suppose now with this data and more sequence that is generated in India for human being, that certain kind of variants are there, and that what genome imprint you have. Okay, instead of having your horoscope, you have a genoscope. And what you can tell this know that if I marry with this person with the similar genetic profile or the very what will happen if certain disease is getting inherited in the family. Okay, how much more diverse individual I should marry. Okay, and if even after that, uh, if I conceive, what kind of genetic variations or variants can be created. Okay, and these would significantly help. Okay, this such counseling from the clinicians and the researchers would very effectively help to to limit such such disease predispositions, and also help in building a more diverse genetic pool, and therefore have a better life. And in such a way, more and more disease can get diluted, and so on. So coming back again to this Genome India and beyond, where I started, I already discussed how all these aspects are very important and very essential in order to understand, and how these sequences or this study can go to a next level or next generation's study level, where it would be very important and essential for all of us to understand what kinds of genes we harbor, and what kind of disease predisposition we have, and how it would affect our life or lifestyle, and so on. So with this take home message, I would like to thank you for your attention, and I would take any questions that you have, and so on.
Thank you so much, sir. We have actually many questions. We will take few of them.
Sure, go ahead.
Yes, sir. So, you discussed about the role of artificial intelligence in genome analysis. Are we as Indians developing any software to decode the data that we have got?
Sorry, I missed the last part of the question. Can you please repeat it?
So, are we as Indians are developing any software to decode the data that we have gathered through this?
Oh yes. This is an ongoing process. There's such experiments or such aspects are being studied at multiple level, from a very lower level to very high level, and this is already in process. It's in progress.
Okay, sir.
Absolutely.
Thank you, sir. There is another question regarding the platforms used for sequencing that you mentioned. Is one DNA sequenced using all these platforms that you have mentioned for better accuracy?
Yes. So what happens is, like when I told you in my in the talk that I discussed, that uh, that certain component regions of genomes becomes difficult to sequence, and so on, right? Or so the short read versus long read. So when there is a new genomes, when you have to map it, you tend to lose certain regions. Okay, that's where what people do is the hybrid assembly, where both short read and long read assemblies can be integrated together to make to stitch the whole genome. Okay, so this is being regularly done. If you get a very good, read, then it is fine. But if you don't get, then mostly, both long read and short read sequencing are integrated together.
Okay, thank you so much, sir. There is one more question regarding how the Genome India project can help in Indian agriculture.
In Indian agriculture,
Yes, sir.
Genome India project can help in Indian agriculture. A very good question. From human to agriculture, I suppose it would certainly help in a way where you would know what people are allergic to. Okay, some people are allergic to certain things like peanuts or egg yolks or brinjal and so on, and if there are genetic components into that. Okay, so one can know from these genes that what they should be eating, what they cannot be eating. Okay, if certain community is growing certain kind of agriculture, like if you go in villages and all, okay, they might be growing certain kind of aspects, so that can be kind of integrated, which I think would be important in terms of allergy, in terms of digestion capacity, and so on. So it will certainly give us important information.
Yes, thank you, sir. And we can also take one last question. Are there any simultaneous studies going on with respect to epigenome?
With respect to
Epigenome.
Uh, with genome?
Epigenome.
Oh yeah, yeah, yeah. So I am myself working on epigenetics and so on. That's my basically work. So genome and epigenome works together actually, or they also work separately. Okay, where if you have a genetic component, say for example, we are harboring certain mutations for certain cancers, but we are not suffering from cancer, right? So there is the epigenome which actually affects. Okay, say for example, epigenome is very dynamic. There have been studies that a person doing regular exercise versus person doing very less exercise. If you take their muscles and look into their genome, the person is who is doing regularly exercise, they have more active genes, they have much less methylated genomes compared to the genomes of the person who is or the muscle genomes, which is basically for an ideal person. Okay, so epigenome is playing an extremely important role in almost all your aspect as to in day-to-day life, and that's where epigenome and genomes have to be integrated together in order to understand what it has. Okay, suppose you have a gene that is mutated. Okay, and if that gene is mutated and expressed, you will have a cancer. But suppose you have an epigenome, but that gene is actually or is highly methylated, then that gene will not be expressed, right? And we will not have cancer. So even though you have a mutation, but because of high DNA methylation over there, or histone methylation, or the repressive marks, those genes are not expressed. And now when suddenly something happens in your lifestyle, something changes in you, and then that repression is removed, and then you develop cancer. So these there are multiple examples like this which is present, and therefore the integration of the genomes and epigenomes are very important to understand the gene regulatory complexities.
Right. So there is one more question. I know we are running out of time, but I'll take that. Our students were interested to know on what criteria do we select the participants from genome analysis, and how do we maintain the confidentiality of the data?
Okay, so I'll answer the second question first. The confidentiality of the data by rule, it becomes randomized. Okay, no one knows any information about, no participants know any information about which individual, and so on. So government has come out with very stringent rules and regulations in order to really control this, or what I would say is a more of a stringent ethical practices. Okay, in this. Now the first part of question is, we when so there are multiple aspects that a person can participate in. They If they have a very good computation knowledge, or very good programming aptitude, or the person is more inclined towards bioinformatics, that can they can work. The person who's working in pure biology can also work in
Sir,
order for
actually,
sample
not for the
management.
analysis, they were interested to know the participation as in for giving the sample for the project.
Okay, okay. Right now, too, they have collected 10,000. There is no, you know, funds or projects as of now. But there is already, there could be some project that will come out in future where participations will be invited, and so on. And so what happens is this: the group discuss or understand the genetic diversity in the India, and based on that, the population is decided. Okay, so that first you understand the more homogeneous population, then you go for more heterogeneous population.
Okay.
So that is how it would work.
Okay, thank you so much, sir. That was a great presentation and you took us from very bottom of the very basic concepts of the genetics to the Human Genome Project and its future as well. Thank you so much. I express my sincere thanks for taking out time and joining us here. Thank you.
Thank you very much for having me, and I hope it would benefit the students. I would take leave now. Thank you very much.
Science is best understood when presented in engaging and creative format. Up next, we have an exciting street play by our talented students, shedding light on significance of Genome India. Let's sit back and enjoy. होते हैं, मतलब लाल रक्त कोशिकाएं, उनमें हीमोग्लोबिन की असामान्यता के कारण ये रोग होता है सिकल सेल एनीमिया।
कोई सीरियस बीमारी है क्या डॉक्टर?
डॉक्टर प्लीज।
अरे सॉरी, कोई बात नहीं।
ये पूछिए। एक मिनट, क्या ये बीमारी आपकी बेटी को हुई है?
हाँ, ये बीमारी मेरी बेटी को हुई है। ये कुछ खाती पीती नहीं है ना, इस वजह से।
नहीं, आप गलत समझ रहे हो। ये बीमारी की वजह ये है ही नहीं।
आपको कैसे पता है बीमारी की वजह ये नहीं है? आप हो कौन?
मैं एक साइंटिस्ट हूँ और मैं इस बीमारी पर रिसर्च कर रही हूँ। चलिए मैं आपको अपने मैजिक लैब की सैर करवाती हूँ। मैजिक लैब, ये क्या होता है? आइए देखते हैं। तो देखिए, ये है मेरा मैजिक लैब। बेटे, आज बॉस ने एक नए आदमी को लेकर आया है, जरा देखते हैं। हाय, क्या आप जानते हो मैं कौन हूँ? फिर से चालू हो गया तेरा? एक ही बात मत सुनिए।
अरे तुम तो बड़े अजीब दिख रहे हो। तुम कौन हो?
मैं बताता हूँ मैं कौन हूँ। क्या आपकी बेटी है क्या?
हाँ।
आपकी नाक और आपकी नाक बिल्कुल सेम है। पता है क्यों? मेरी वजह से, क्योंकि मैं हूँ क्रोमोसोम, यानी कि मेरा नाम है क्रोमोसोम, और ये जो नाक सेम है ना, वो मेरी वजह से है। क्या रे क्रोमोसोम, ज्यादा ही पटेली मार है। तू जो यहाँ खड़ा है ना, वो मेरे और मेरी चार पे स्पेल की वजह से खड़ा है, और तू जो ये बोल रहा है ना नाक सेम है, वो तेरे नहीं मेरे वजह से है। डीएनए के आगे कोई बोल सकता है क्या? डीएनए भाई। हाँ, मैं बोल सकता हूँ। जीन नहीं तो कौन बे? हाँ, जीन नहीं तो कौन बे? है कौन इधर? जीन नहीं तो कौन? ये जो तुम लोग बोल रहे हो ना, नाक सेम है करके, तुम्हारी वजह से, वो मेरी वजह से है। मैं ट्रांसफर हुआ तभी उन दोनों की नाक सेम है। बड़े हैं यहाँ बोलने वाले, जो मैं बोलूंगी वो तुमको सुननी पड़ेगी, जैसे कि अभी तक हमारी बहुत चल रही। चलो अभी शांत हो जाओ तुम लोग। आया बड़ा डीएनए, आया बड़ा क्रोमोसोम, चलो भागो तुम लोग।
अरे कहना क्या चाहते हो?
रुकिए, मैं आपको विस्तार से समझाती हूँ। तो देखिए, ये है डीएनए और इसका एक सेगमेंट है जीन, और हम इसे थोड़ा और कंडेंस करें तो ये बन जाता है क्रोमोसोम। देखो क्या पेड़ है, चादर ओढ़े या खड़ा कोई। कौन है जिसने मेरी ये नींद खराब कर दी? इतने अच्छे से सोया था मैं।
अरे जैसे मेरे और मेरे पापा की नाक सेम है, वैसे आप भी उसकी तरह ही दिखते हो, लेकिन आप थोड़े से कमजोर क्यों लग रहे हो?
एक्सक्यूज मी, आप मेरा नाम इससे जोड़ रहे हो? मैं कहा हेल्दी क्रोमोसोम, और ये तो रोगी है, रोगी। मैं कहा ये कहा। बॉस, क्रोमोसोम इज अब्यूजिंग मी। नो, इट इज नॉट अब्यूज, ही इज टेलिंग अ लाई। तू चुपचाप जाके बैठा है ना, मार खाएगा।
क्या पहले से ही ऐसे हो?
नहीं, भोला भाला था, सीधा साधा था, मैं तो नादान था। दुनियादारी से, होशियारी से, मैं तो अनजान था। लगी चोट ऐसे मेरे भोले दिल पे, बिखर सा गया टूट के। मैं भी पहले इनके जैसे हेल्दी क्रोमोसोम था, पर मेरे डीएनए के बेस पेयर में कुछ चेंजेस की वजह से मैं ऐसा हो गया हूँ। आपकी बेटी को जो सिकल सेल एनीमिया हुआ है, उसके लिए भी यही रिस्पॉन्सिबल है। मारो इसे, मारो।
ये जेनेटिक डिसऑर्डर क्या होता है?
जेनेटिक डिसऑर्डर एक ऐसा डिसऑर्डर होता है जो जेनरेशन टू जेनरेशन ट्रांसफर होता है।
पापा, तो हमारे घर में जो न्यू बेबी आने वाला है, क्या उसको भी ये डिसऑर्डर होगा?
हाँ, हो सकता है।
फिर तो ऐसी कई सारी बीमारियां होती होंगी जो जनरेशन टू जनरेशन ट्रांसफर होती है, और हमें कैसे पता चलेगा?
क्यों नहीं पता चलेगा? ऐसे बहुत सारे साइंटिस्ट हैं जिन्होंने मिलकर एक प्रोजेक्ट पर काम
मिनट में डीएनए के सीक्वेंस को रीड कर सकते हैं और इससे हम डिजीज कॉजिंग जीन को भी आइडेंटिफाई कर सकते हैं। ह्यूमन जीनोम प्रोजेक्ट, ये प्रोजेक्ट एक वर्ल्ड वाइड प्रोजेक्ट है। ये 1990 में स्टार्ट हुआ था और 2003 में कंप्लीट हुआ था। इस प्रोजेक्ट के अंदर बहुत सारे देशों का कोलैबोरेशन भी रह चुका है।
ये प्रोजेक्ट के अंदर बहुत सारे ह्यूमन जीन के लोकेशन और स्ट्रक्चर और उनके ऑर्गेनाइजेशन को आइडेंटिफाई किया गया है। हमारा जो ह्यूमन जीनोम है वो बहुत ज्यादा कॉम्प्लेक्स है, 3 बिलियन बेस पेयर से बना हुआ है।
मैं हूँ डीएनए, छोटा पैकेट बड़ा धमाका। क्या आपको पता है आपके शरीर में मेरे जैसे 3 बिलियन बेस पेयर है? 3 बिलियन? अगर ये बच्ची इन 3 बिलियन बेस पेयर को अकेली रीड करने चाहती तो इसकी तो पूरी जिंदगी निकल जाएगी। इसलिए दुनिया भर के साइंटिस्ट ने मिलकर इस प्रॉब्लम का सलूशन निकाला है। अभी जीन को पढ़ तो लिया, पर उसका क्या फायदा? क्या फायदा? इस प्रोजेक्ट के थ्रू हम जीनोम सीक्वेंसिंग की हेल्प से बहुत सारे जेनेटिक डिसऑर्डर को आइडेंटिफाई कर सकते हैं, जैसे कि सिकल सेल एनीमिया और एपिलेप्सी, यानी कि मिर्गी और पार्किंसंस, मतलब कि हमारे ब्रेन से बॉडी का कनेक्शन हट जाना। आपको समझा क्या? आपको समझा क्या? मुझे भी नहीं समझा। आखिर क्या चाहते हो? ये जीनोम सीक्वेंसिंग आखिर होता क्या है? बताओ। रुको जरा, सब्र करो, मैं बताती हूँ जीनोम सीक्वेंसिंग होता है। जीनोम सीक्वेंसिंग यानी कि किसी भी ऑर्गेनिज्म के कंप्लीट डीएनए को रीड कर लेना, यही होता है जीनोम सीक्वेंसिंग। वाह, ये तो कितना इंटरेस्टिंग है। मुझे तो इसके बारे में कुछ पता ही नहीं था। मुझे भी ये सब पढ़ना है और इनके जैसे बड़ा साइंटिस्ट बनना है।
और अभी तक तो इस प्रोजेक्ट पर सिर्फ बाहर के साइंटिस्ट ने काम किया है और इंडिया में ये सब फैसिलिटीज कहाँ?
अरे ऐसी बात नहीं है, हमारे इंडिया में कई साइंटिस्ट ने इंडियन जीनोम प्रोजेक्ट की वजह से 10,000 डीएनए सीक्वेंसिंग करने का गोल पूरा किया है और इसमें डिफरेंट एथनिसिटी के लोगों के डीएनए की सीक्वेंसिंग हुई है। आप ही सोचिए, अगर हमें बीमारियों के बारे में पहले ही पता चल गया तो हम इसे डिटेक्ट करके मेडिकेशन के थ्रू ठीक कर सकते हैं। हमारे इंडिया में द सेंटर ऑफ सेलुलर एंड मॉलिक्यूलर बायोलॉजी, जो हैदराबाद में स्थित है, एंड द इंस्टीट्यूट ऑफ जीनोमिक्स एंड इंटीग्रेटेड बायोलॉजी, जो दिल्ली में स्थित है, इन दोनों ने मिलकर ह्यूमन जीनोम के सीक्वेंसिंग में बहुत बड़ा योगदान दिया है। हालांकि यही नहीं, जो बीमारियां पहले ठीक नहीं हो सकती थी, जैसे कि कैंसर, डायबिटीज एंड कार्डियोवास्कुलर डिजीजेस, इनका भी निदान निकल चुका है और ये सारी चीजें पॉसिबल हुए हमारे इंडियन ह्यूमन जीनोम प्रोजेक्ट की वजह से। इसलिए कहते हैं, अगर स्वस्थ रहेगा इंडिया तभी तो आगे बढ़ेगा इंडिया।
We would love to hear from you. If anyone would like to share your feedback for today's session, please feel free to come on the stage and anyone else? Good afternoon, everybody. Good afternoon, ma'am. Good afternoon, sir. Thank you so much for giving us this opportunity. I would really like to thank our HODs to give this GenomeIndia project for us as a field project. Firstly, at FY level, I would really like to thank you from all our heart and from our FY, SY and TY that you have really given us an opportunity which we really appreciate. When this field project was introduced to us, our attitude towards it firstly was a very different attitude. And over the time that we have been reading about this project, we have been so involved. Personally, we feel that we have been a part of this project which is a government initiative. Since morning, everybody is on their toes, and we are feeling that this is like an event that we have at our home, which is a very personal event to us. And everybody is working hard, and we would work hard this way. Um, Thank you so much for. I would really like and appreciate the efforts that the students have taken, and we will really make this a huge success. Thank you. I would also request everyone to provide the feedback by scanning the displayed QR code. As we conclude this inaugural session, it is time to express our gratitude. I request Dr. Jayashree Pawar to deliver the vote of thanks. Thank you, Nikita. It's really an honor for me to propose vote of thanks in this elite gathering of SVATV celebrating the success of the ambitious GenomeIndia Project. I express sincere gratitude to today's eminent speakers, the scientists who are actually involved in the GenomeIndia Project, Dr. Thangaraj and Dr. Punit Prasad, who, in spite of their very busy schedule, kindly accepted our last-minute invitation for this event. So, your speech has not only provided valuable insights on GenomeIndia, but also motivated all our students for career in related fields. Thank you very much. We cannot thank enough Vidya Prasad Mandal for all the unconditional support always provided. I wish to thank Dr. Mahesh Vedekar sir, Honorable Member of DPM, who actually suggested us celebrating the success of Genome India in such a unique manner. So thank you very much. The entire department is thoroughly enjoying the fest, as we could see when Nishigandha spoke about it. Right from deep reading on the project to organizing poster and model presentations, designing the games and street plays, which have been the innovative, fun learn ideas by Dr. Kalpita. We could notice hidden talents in our own students during the activity. Thank you very much, sir. I wish to thank our dynamic principals, Dr. Vinda Mansalamkar ma'am, Principal B. N. Balwadkar College of Science, and Dr. Nayak sir, Principal BPS College of Engineering, for constant guidance and support. We also would like to express our gratitude to principals on the campus, Dr. Naik ma'am, Principal Joshi Medical College, Dr. Joshi sir, Principal Briggs, and Dr. Srividya ma'am, Principal TNC Law College, for all their valuable suggestions. Thank you to our sponsors, Appendix India Private Limited and Rotary Club of Thane Suburban, for the financial support provided. A big thank you to Dr. Patil sir and Dr. Vasse sir from Medical College for all the guidance and unconditional support that has been provided. Thank you, Mr. Siddharth, for the technical support, Mr. Joshi and his team for the panel arrangements. Thanks are due to many people working hard behind the curtains, staff and students, our all the time enthusiastic volunteers of our department, as well as the VPN College of Engineering for the models, teaching and non-teaching staff of our department, biotechnology and microbiology. They have been working very hard for last few days, and because of that hard work, we could see all this, all the presentations and lectures that have been going on smoothly, that we could witnessed today. So thank you so much, all of you. Special thanks are due to Saili ma'am and Purvi ma'am for all the online arrangements, the beautiful video of national anthem that filled our hearts with pride. Last but not the least, thank you so much, the wonderful audience. Thank you so much. Thank you. The session is concluded. I